In this presentation, I'm going to talk about importance of sample preparation before HPLC, filtration in pharmaceutical analysis, some of the properties of membrane filters and devices, various means of improving throughput, data accuracy and reproducibility. And finally, at the end, after Ahmed's presentation, we'll do some questions and answers. There are three key components of success in HPLC and LC-MS analysis. One is HPLC columns. The second one is mobile phase components. And the third one is sample and mobile phase preparation. In this seminar, we are going to focus on the sample and mobile phase preparation. Why is sample preparation important for HPLC? Because without sample preparation, samples can become incompatible for HPLC or UPLC or LC-MS analysis. Many a times there are dissolved particles in the sample or mobile phase that can clog or reduce life of chromatography. Some of the sample matrix components can also have impurities, which make chromatograms very challenging to interpret. For example, what we have shown at the bottom of the slide, on the left-hand side, you have a raw sample, which is not being gone through a preparation stage. And on the right-hand side, you have a clean sample, wherein majority of the impurities have been taken out of the sample, leaving behind the analytes of interest. So essentially, sample preparation removes particles, contaminants, and interferences from the sample and brings sample to a form amenable to downstream analysis. In this table, we are showing various sample preparation techniques and what they offer. As you go down the table, from filtration all the way to SPE or solid phase extraction, higher and higher amounts of components or matrix impurities can be removed using these sample preparation technique. The table also refers to what type of samples can be used, its level of complexity, and the cost associated with it. Filtration is one of the simplest sample preparation techniques, but it only can be used for removal of particles when they are present in the sample. As you go down the table, the complexity of sample preparation goes up, but at the same time, the cost of sample preparation also goes up. Depending on the sample and the matrix complexity, you will have to select the appropriate sample preparation technique. Now, going to filtration in pharmaceutical analysis, I'm going to focus mostly on the pharmaceutical analysis here. Especially in pharmaceutical QC testing, there involves a lot of assays which require filtration. For example, drug dissolution testing is performed on every oral solid dosage form, and all the samples are filtered prior to analysis. There are also a large number of dissolution tests conducted each year. There are various other pharmaceutical QC tests that require filtration for sample clarification. For example, assay, content uniformity, stability and shelf life, etc. And in most cases, HPLC or LC-MS is the downstream analysis for all of these assays where removal of particles before analysis is critical. Focusing on drug dissolution testing, which is one of the most commonly used pharmaceutical QC tests, why is sample filtration important in drug dissolution testing? It's because filtration stops the dissolution process. The purpose of dissolution testing is to find out how much of the active pharmaceutical ingredient or API dissolves at a certain time point in the dissolution media. So the dissolution medium contains both the dissolved and the undissolved inert ingredients as well as the active compound. Filtration removes the undissolved ingredients, thus providing a representative sample which can be used for quantitation of API at a certain time point. For filtration in dissolution testing, there are two types of common sample preparation methods or common sample filtration methods. One is using syringe filters and the other one using the frit filters. Syringe filters have a pore size of typically 0.1 to 1 micron, whereas the frits have much larger pore size. Their pore size distribution is also narrow compared with the frit filter. And their pore size rating is based off of largest pore as opposed to the average pore size for frit filters. In dissolution testing and any HPLC analysis, syringe filtration is necessary when the downstream analysis is HPLC. Over the years, I have worked with various customers doing HPLC, dissolution testing, as well as various pharmaceutical QC analysis. Some of the common challenges that customers face or researchers face when they have to filter samples for HPLC analysis is something that a filter can filter sample directly into HPLC vial can prevent multiple transfer steps. Some of the filter attribute that this causes is the device configuration. When I started doing these seminars, UHPLC was new on the space, and people were concerned whether the same filter that they were using for HPLC analysis can be used for UHPLC or not. Customers in dissolution labs were using frit filters because many a times they were automation compatible or they came with the system. But customers were also concerned that it would be good to have frits which are absolutely pore size rated. Because as I said earlier, frits usually have an average pore size. The most important thing in customers' mind was that filters show variable drug binding and extractables. Both of these impact quantitation of analyte in the downstream assay. So filter without either of those would be great. So the bottom line question was, are we using the right filter if I choose a filter that worked last time with compound X, which is very common practice in the labs. So even though customers and researchers have the questions about how to choose a syringe filter or what I need to look for in a syringe filter, when we ask them how do they actually choose the syringe filter, the top answer is always what is in the lab drawer or in the stockroom. Oh, I always use the yellow filter that is in the lab, or I use the filter that worked the last time. For people who are using automated systems or automated filter changing systems, the filter has to be automation compatible. Sometimes the equipment manufacturers recommend certain filters. For QC labs, especially, the filter that they have to use is written in SOPs, which is validated by the R&D analytical team. Or sometimes it can be also specified in the USP protocols and publications. But the key factors to consider when selecting a syringe filter is by pore size, by membrane material. It must have a pre-filter in case you're filtering samples containing high level of particulates, and its chemical compatibility. And these factors rarely come into discussion when you directly ask customers about how they choose a syringe filter. Many a times, or 80% of the times, the filter that was used for the previous sample or a previous type of material will work for your analysis. But when it doesn't work, then you do not know why it is not working, because it wasn't selected based on the key characteristics. Now in the next few slides, we are going to go over some of the key membrane filter characteristics as well as the device characteristics. I said earlier that syringe. So how do we measure these pore sizes of these membranes? So typically, pore size of a membrane is measured by an indirect technique called as bubble point, where it is essentially a pressure that is required to force air through the wetted porous membrane. The way this test is carried out is the membrane is wetted with a liquid and it is held in a holder. On one side, you have liquid, for example, water, and on the other side, you would have air, which is now pressed against this wetted membrane. There comes a point where there's enough pressure that it allows the air to go from one side of the membrane to the other side of the membrane, creating a steady stream of bubbles. And because of that, this particular test is known as bubble point. Millipore and Merck Life Sciences has been involved in standardization of this test right from the beginning. The other commonly used test that some of you might be familiar with is the bacterial challenge, where certain sized bacteria are allowed to filter through a membrane, collecting the filtrate and analyzing it for presence of bacteria. This test is generally used when you're looking for sterilizing grade filters. In R&D, we also use scanning electron microscopy or porosimetry. For example, scanning electron microscopy is what shows the porosity and the pore structure or membrane morphology. And as you can clearly see, the different membranes that are shown here have very different pore structures. And the pore structure and morphology is determined by the manufacturing process. For example, the PVDF, the polyether sulfone, and the nylon membrane are manufactured by solvent precipitation, where a solution of polymer is precipitated out using a non-solvent. It creates a random network of pore structures. If you look at the polytetrafluoroethylene or PTFE membrane on the right-hand side, top right-hand side, you can see that the pores are elongated in one particular dimension. That is because PTFE membrane starts off as a PTFE tape, which is heated and expanded under pressure and pulled under a certain direction. If you look at the bottom left corner SEM image, a polycarbonate membrane actually has pores which are completely cylindrical, and they go through the membrane in a cylindrical fashion. A polycarbonate membrane is prepared by track etching process, where a polycarbonate film is first irradiated with high energy radiation, followed by chemical etching, where the area which was irradiated can be etched out and leaves behind these pores. Similar to pore morphology, sometimes the pore, the symmetry of pores across the thickness of the membrane is also different. Here there are two examples of a symmetric versus an asymmetric membrane. The membrane at the top is a symmetric PVDF or polyvinylidene fluoride membrane, which has very similar pore structure at the top as well as at the bottom. The membrane shown at the bottom has an asymmetric pore structure, meaning that the pore size at the top of the membrane is slightly larger than the pore size at the bottom of the membrane. Why is the pore size and pore distribution critical? Because particle retention by various syringe filters or membranes is dependent on the pore size and pore size distribution. In this example, we tested syringe filters from two different manufacturers and allowed filtration of 0.3 micron latex bead suspension through a 0.2 micron membrane filter. A 0.2 micron membrane filter should retain quantitatively all the particles that were present in a 0.3 micron latex suspension. But as you can clearly see, for the PTFE versus the polypropylene membranes, as well as the PVDF membranes from the two vendors, only the PTFE and the PVDF membrane from vendor A shows the quantitative recovery of the particles, whereas the polypropylene and the PVDF membrane from vendor B allows for roughly 20% of the particles to go through this syringe filter. Similar behavior was also observed for this polypropylene membrane compared to the PTFE membrane when a mobile phase, meaning a mixture of water and acetonitrile, was filtered through these membrane filters. And then these were allowed to run through a HPLC system overnight. What we did in this case was we filtered 50% acetonitrile water mixture and ran it through HPLC system and monitored the back pressure without any sample injection. As you can clearly see, the polypropylene membrane allows for some particles to go through this membrane filter, causing an increase in the back pressure, even without filtration of, even without addition of any sample injections. Another characteristics of membranes that is important when filtration of sample is hydrophilicity or hydrophobicity of membrane. In this example, we have 3 images of water on 0.1 micron PVDF membrane. For a hydrophilic PVDF, you can clearly see that the water wets the membrane, thereby allowing filtration of aqueous solutions through this 0.1 micron PVDF. On the other hand, if the PVDF membrane was hydrophobic or superphobic, water will not wet this membrane out. When we go to a hydrophobic, when we go to a solvent on the same 0.1 micron membrane, both the hydrophilic and the hydrophobic membranes are wetted out by this solvent. But even in this case, you can clearly see that the superphobic PVDF membrane is really not wetted by even isopropyl alcohol. For pharmaceutical analysis, where most commonly used samples and solutions are aqueous, a hydrophilic membrane is to be used for filtration of samples. Now that we have talked about properties of membrane filtration, properties of membrane filters and devices, let's talk about various means of improving throughput. One of the simplest methods for improving sample throughput and getting more samples filtered in a smaller amount of time, or making filtration easier, is just by choosing the right filter based on the sample volume. If you have a sample volume less than 1 ml, typically we recommend syringe filters which are of the size of 4 millimeter, simply because they have smaller sample hold of volume, and also because they have smaller membrane area, which leads to lower amount of extractables and analyte binding. But on the other hand, if you have large sample volume, like somewhere in the range of 10 to 100 milliliters, we recommend a 33 millimeter syringe filter. They still have a smaller hold of volume, but because it has large surface area, it allows for faster filtration of larger sample volume. And so you can imagine if you had to filter a 15 to 20 ml of sample through a 4 millimeter syringe filter. It will take very long time. The filter might clog up really, really quickly. And on the other hand, if you use a 33 millimeter syringe filter for filtration of 0.5 milliliters or 500 microliters of sample, you might lose a significant amount of sample to the filter itself. And you might see higher level of analyte binding and extractables. Increase the throughput further using syringe filters. Millipore Sigma and Merck Life Science launched a product a few years back called Simplicity, which is vacuum-based sample filtration. It allows for filtration of 8 samples directly into HPLC vials, taking care of some of the sample concerns or device configuration concerns that the customers had raised. Many of us have come across samples which are difficult to filter. These are samples which contain high level of particulates, causing the membrane filter to clog up really, really quickly. Customers use multiple syringe filters to get the desired volume that is to be filtered. Sometimes they use a coarse pre-filter followed by a membrane solution. But a better solution for such samples is using a syringe filter that contains multiple layers of pre-filters, and a final 0.45 or a 0.2 micron membrane can significantly improve the throughput. These syringe filters contain multiple layers of typically glass fiber pre-filters, which trap the large particulates, followed by a 0.45 or a 0.2 micron membrane, which allows for final filtration for sample to be analyzed directly by HPLC. The chart on the right-hand side clearly shows how much sample volume can be filtered by just using a multigrade glass fiber pre-filter in a syringe filter format, followed by a PVDF membrane. Roughly 6 to 8 times higher sample volume can be easily filtered before the membrane clogs up when using a multi-layer syringe filter. Sometimes customers also use automated filter changing stations. This is especially true for large-scale dissolution testing labs or pharmaceutical QC labs. It is important that the filters that are chosen are compatible with automation and that the data is not influenced by automated sampling. Within our portfolio, we have various formats of syringe filters available for filtration using automated filter changing station. They go from a membrane-only filter all the way to a multigrade glass fiber pre-filter followed by the membrane filter. Occasionally, we come across samples which are viscous. They do not contain large load of particulate material, but they have some of the high viscosity polymeric materials that are dissolved in the sample, which make it extremely difficult to filter them using a traditional syringe filter solution. We also offer a variety of solutions for filtration of samples that are viscous in nature. We have ultrafiltration membranes containing regenerated cellulose as a low binding membrane, allowing for filtration of samples which contain dissolved impurities. The separation here is based on the molecular weight and the molecular weight difference between the analyte of interest, which is generally a low molecular weight pharmaceutical component, and high molecular weight polymers, which are used as excipients in these formulations. These devices are commonly used for protein concentration and desalting, as well as DNA concentration and desalting. But these can be easily used for removal of polymeric excipients when we come across solutions containing dissolved polymeric excipients. Now that we have talked about membrane characteristics as well as means to improve throughput, we are going to the main part of the seminar, which talks about the data accuracy and reproducibility. Because at the end of the day, as analytical scientists, we are all interested in making sure that our data is accurate and reproducible. When we look at data accuracy and reproducibility, there are three characteristics that we have to take into account. The chemical compatibility, because that maintains the membrane integrity and ensures consistent and reproducible performance. Secondly, variable drug binding or analyte binding, which are dependent on the characteristics of the analyte, properties of membrane and the prefilter, and they are also concentration dependent. And extractables, which are soluble materials that elute out of a device during filtration. These extractables are also based on chemical compatibility of the device and the solvent, and they can affect the downstream analysis. Looking at chemical compatibility, this table shows chemical compatibility of commonly used membranes like PTFE, PVDF, nylon, PES, and regenerated cellulose, as well as common housing materials for syringe filters, the HDPE and polypropylene, across a wide range of solvents. As you can clearly see, of the membrane materials shown here, the PTFE membrane has the broadest chemical compatibility across all the types of chemicals. The PTFE membrane shown here is hydrophilic in nature. Even though commonly used PTFE membrane is hydrophobic, within our portfolio, we have developed a process to hydrophilize the PTFE membrane to give it compatibility for organic solvents, but also allow for filtration of aqueous solutions through the same membrane. So the hydrophilic PTFE membrane depicted here is compatible with organic solvents as well as aqueous solutions. Now going to analyte binding. Since our main aim for doing any HPLC analysis is generally quantitation of analyte, any analyte binding to the membrane filter will cause a reduction in the quantitation of analyte, which means we will not get the right concentration of analyte in our final sample. Here we are showing results from filtration of samples using two different syringe filters, the nylon syringe filters on the left-hand side and the hydrophilic PTFE syringe filters on the right-hand side. The way these experiments were done, we selected 4 different oral formulations, ranitidine, acetaminophen, ibuprofen, and loratadine, and carried out drug dissolution testing for these formulations using standard USP protocols. After the dissolution test was completed, samples were filtered using either nylon syringe filters or the PTFE, hydrophilic PTFE syringe filters. And various fractions were collected, starting from the first ml of fraction all the way to the 10th ml of fraction. The filtrate was analyzed for quantitation of API, in this case, ranitidine, acetaminophen, ibuprofen, and loratadine. And as you can clearly see, for the nylon membrane or the nylon syringe filter, the first ml of filtrate shows significant binding of analyte, leading to low recovery of analyte in the first ml of filtrate. On the other hand, when you use hydrophilic PTFE-based syringe filter, there is no analyte binding observed even for the first ml of the filtrate, and quantitative recovery can be obtained. We also looked at dissolution testing of a multi-component tablet and filtered this, following dissolution test, filtered this filtrate using either hydrophilic PTFE, nylon, or PVDF membrane. For acetaminophen as one of the components in this formulation, you can clearly see that only 58% of acetaminophen was recovered for the first ml of filtrate, whereas both the hydrophilic PTFE and the PVDF membranes show quantitative recovery. Similar results were also observed for acetyl salicylic acid, which was part of this formulation, where only 28% recovery was obtained for first ml of filtrate. On the other hand, for caffeine, which is a neutral molecule, there was no analyte binding seen for various different membranes, whether it was PTFE, nylon, or PVDF. As I said earlier, the analyte binding is dependent on the analyte itself, its physicochemical characteristics, as well as the membrane and its physicochemical characteristics. So why does nylon membrane show higher level of analyte binding? The reason being, nylon membrane is a polyamide, which means it contains an amino group at one end and a carboxylic acid group at the other end. And it also contains amide linkages. Majority of our APIs are either acidic or basic. Actually, 60% of APIs that are used in pharmaceutical world are basic molecules, meaning having a pKa greater than 7. So under ideal condition, these acidic or basic molecules can be charged and can form electrostatic interactions with the nylon membrane. Similarly, the amide bonds can form very strong hydrogen bonds with various analytes. Thereby, these two reasons cause the nylon membrane to show strong analyte binding to various analytes. Both the PTFE and the PVDF membranes do not contain various functional groups or very little functional groups. And because of that, they generally tend to show very low analyte binding, even for the first ml of filtrate. If you're working with proteins as analytes, you also have to take into account the protein binding of various membranes. Within our portfolio, we have two different membranes, a polyether sulfone, as well as the hydrophilic PVDF membrane, which have very low protein binding. Looking at membranes from different competitors, different membranes tend to show different levels of analyte binding, going from a very, very low level to a very high level. So especially for customers who are working with antibodies as their analytes, they need to take into account whether the membrane that they're using for filtration leads to any analyte binding for the proteins. Now moving on to extractables. First and foremost, let me show you some extractable chromatograms for the common housing materials that are used for syringe filter manufacturing. On the left-hand side, we have HPLC UV extractable chromatogram for a methanol blank and the polypropylene and the HDPE housing. On the right-hand side, we have LCMS spectra or mass spectra for extractables for an acetonitrile blank or HDPE housing. The way these experiments were done, a sample or a solvent was filtered through empty polypropylene or HDPE housing, and the filtrate was then analyzed by reverse phase chromatography using 0 to 100% acetonitrile gradient. As you can clearly see, the methanol blank and the polypropylene housing or HDP housing do not show any additional extractable peaks. And same is the case in the mass spectrum as well. The peaks that were seen in the blank are the only peaks that are seen from the HDP housing. Here is an example of extractables from a nylon syringe filter, which can cause problems in the downstream HPLC analysis. When water was filtered through the nylon membrane and the filtrate was analyzed using similar reverse phase chromatography, you can see 3 or up to 4 chromatographic peaks right in the middle of this chromatogram. These peaks can cause problems in downstream analysis. If these peaks are well separated from analyte of interest, the quantitation of analyte may not be impacted, but if you are submitting these results to any regulatory bodies, these additional peaks have to be explained and quantified, and in some cases even have to be identified. If these peaks elute along with the analyte of interest, then quantitation of analyte becomes complicated because you never know what is the level of these extractables from sample to sample. Generally, extractables can be eliminated by pre-rinsing the device with 3 to 5 ml of solvent or sample. Here is as an example of various HPLC certified syringe filters from various vendors, which went through the same extractable process using 70% acetonitrile and 30% water as an extraction solvent. The polypropylene syringe filters from vendor B and the nylon syringe filters from vendor C, as well as the hydrophilic PTFE-based syringe filters from vendor A. These are all certified for HPLC using HPLC UV. But when we did extractable testing using our protocol, you can clearly see that all of these HPLC certified membranes and devices still tend to show high levels of extractables, except for the hydrophilic PTFE from the vendor A. So it is important to understand that syringe filters, even HPLC certified syringe filters from different vendors can have different levels of extractables. Earlier, I talked about the Amicon Ultra devices that can be used for removal of polymeric excipients. Here is an example of extractable chromatogram from using a regenerated cellulose-based Amicon ultrafiltration device and comparing it against various ultrafiltration devices available from different vendors which contain polyether sulfone membrane. As you can clearly see, the extract coming out of an Amicon Ultra device is almost as clean as the blank, whereas the extracts coming out of the PES-based ultrafiltration devices has higher levels of extractable peaks, which can cause problems in downstream HPLC analysis. We also looked at LC-MS because LC-MS is growing in popularity and use across industry as well as academia. And majority of the HPLC certified syringe filters are certified by HPLC UV. So we looked at 3 different membrane filters: hydrophilic PTFE, polypropylene, and nylon membranes. And we looked at various solvents and solutions, mass spec certified whenever available. If you look at the table at the bottom, we gave a qualitative assessment to reproducibility. If you look at batch to batch reproducibility from one vendor to another vendor, we looked at the qualitatively level of extractables as well as the nature of extractables. As you can clearly see, the hydrophilic PTFE syringe filters from Millipore Sigma, which are certified for HPLC by HPLC UV, have a high reproducibility across different lots of membrane filters. Generally, the extractable levels are low, and the nature of extractables are some discrete peaks. So even HPLC certified syringe filters can show higher levels of extractables when we are using them for LC-MS analysis. So it's important to understand that the filters from different vendors can be different and can lead to differences in extractables and analyte binding behavior. Just to show an example of the mass spectral trace coming from a hydrophilic PTFE syringe filter on the left-hand side versus a polypropylene syringe filter on the right-hand side, you can clearly see that the polypropylene syringe filter has much higher level of extractable impurities, as can be seen by various peaks in the mass spectrum. So a suggestion would be to start with hydrophilic PTFE membrane when developing a new method. Many times we blame our syringe filters for extractables and analyte binding, but the syringes that we use for filtration of sample can also cause or add contaminants to the sample. This is an example actually from a customer where they were looking for zinc analysis using ICP analysis. And they saw that plastic syringes with the black piston seal can lead to higher level of zinc contamination in the sample depending on the time of exposure. So in summary, filtration is very simple sample preparation technique, but it is very little understood. Hopefully I have shown data to give you confidence that the hydrophilic PTFE membrane that's part of our portfolio offers very broad chemical compatibility, and it also has very low extractables and analyte binding. Second choice for membrane selection would be a hydrophilic PVDF membrane because it is compatible with most aqueous solutions. It also has low extractables and drug binding, and it is commonly recommended for protein analysis. Finally, select your filtration format based on your throughput needs. Select appropriately sized filter depending on the sample volume, simplicity for vacuum filtration of multiple samples, devices with pre-filters for particle-laden samples, ultrafiltration devices for removal of dissolved polymeric impurities, and automation-compatible filters for filter changing stations. With that, I would like to end my portion of the talk, and I would like to transfer it over to Ahmed, where he's going to talk about mobile phase and water and its importance in HPLC analysis. Ahmed?
Hello, everyone, and thank you, Dr. Vivek, for your nice slides. Today, I'm so happy to share my experience talking about the water as a key reagent in each and every laboratory. The agenda for today, I have only 10 minutes to introduce all the available information that we can talk about in the HPLC application. So the agenda will be talking about introduction on water contamination, type of purified water, standards and specs, laboratory water types and applications, impact of pure and ultra pure water in HPLC application, and the effect of contamination. Merck Water Purification Solutions will be also the last part where I will be closing my slides with. So whenever we are talking about water and ultra pure water, we should be expecting in tap water, which is H2O with some stuff inside. Here, we mainly focus on 5 types of contaminations. Ionic contaminations such as cations, sodium, calcium, anions like chlorine, sulfate, organics, which is naturally like tannic acids or humic acid, or it would be artificial like drug residues or herbicides. Particulates and colloids like sand, dust, silica, microorganisms and byproducts such as bacteria, endotoxins, proteins, enzymes, and nucleases. And also dissolved gases could be part of these contaminations such as oxygen, carbon dioxide, and nitrogen. Whenever we are talking about ultra pure water, it's H2O and nothing else. Second part we need to define what exactly we do mean by highly purified water. Purified water is used in every laboratory and can be nominating using many names as distilled water, deionized water, DI water, ultra pure water, or even 18 mega ohm water. Accordingly, we always need to look after what standards we are following inside our lab. Here is a summary of different types of standards like ISO, ASTM, NCCLS, and even pharmacopoeia, which is defining the several types of water, of highly purified water. For example, if we look at the ISO, they have different names like grade 1, grade 2, grade 3. The ASTM will say no, we are talking about type 1, type 2, type 3, and type 4. NCCLS the same, and pharmacopoeia, here we will talk about European pharmacopoeia and United States pharmacopoeia. As the main monitoring parameters, you will see the definition of resistivity or conductivity. And when we talk about ISOs, you will see the grade 1 starting with 10 mega ohm, which is equivalent to 0.1 microsiemens. ASTM, or American Society for Testing and Material, will be speaking about 18 mega ohm, which is equivalent to 0.056. One more parameter you will see is the pH, which I have seen a lot of people inside the labs are trying to testing the pH with the highly purified water, which is not possible because the pH testing will be based on the mobility of the ion. And when we are speaking about highly purified water, we are speaking about water without ion inside. So, as a summary, most of the laboratory water specification have defined 3 types of water to comply with most and every standards of quality. Type 1, starting from 18.2. When it comes to TOC, we are speaking about less than 5 parts per billion. Particulates, we are speaking about less than 1 unit per milliliter. Bacteria is tested by colony forming unit, and it should be less than 1 colony forming units per 10 milliliter. Of course, we have 3 types, and each type will be reflecting or linked to several types of application. When we are speaking about type 3, we are speaking about pure water with final quality subject to the incoming water quality. The basic lab needs here are non-critical rinsing, heating baths. Type 2, we are speaking about pure water, but with consistent water quality. The standard application for that will be preparation of buffers, microbiology media, glassware rinsing, feeding to clinical analyzers. And here we are speaking about the type 1 or the ultra pure water, which is targeting contaminant removal. And it is mainly linked to the critical applications like HPLC, LCMS, ICP-MS, GC-MS, molecular biology. And it's also important to have an idea on what is the journey where we are purifying the water through different stages of purification. Starting from pre-treatment pack, which is removing the contaminants that can damage the system like chlorine, silica, and large particles. Second, we are using the RO membrane, which is offering us type 3 water because it's removing 95 to 99% of the ion, followed by the EDI or the electrodeionization, which is removing all charged ions to a level of type 2 water. After the EDI, we will have the UV lamp at 254 nanometer, which is killing bacteria until we storing the water in safe conditions so that of 185 nanometer, which is also relating to killing or removing the organics. Then we will have a final purification pack to removing contaminations, followed by the final filter, which is point of use filter, and it's coming with 0.2 micron membrane. Very fast, the rule of water in liquid chromatography is used for mobile phase, preparation of the sample standards, washing and rinsing of the sample and reagent containers, and also for the planks. It is very important to use water of high quality to avoid interference with the measured analytes, as well as to maintain the instrument working in proper condition. Just fast questions, and I will answer myself. What is the major components of the HPLC or LCMS, which may be affected by the contamination coming from the pure water used? Almost every and each component of the HPLC may be affected, like the pump, injector, column, and even the detector. What are the damages we are expecting from each contaminants, like particles? We can see damage to the HPLC and UHPLC pumps and injector, plugging the column and threads, increasing the back pressure. Bacteria may have a major effect on plugging the column and threads, which you can use on the column, increasing the back pressure, releasing organics resulting in ghost peaks. Organics contaminations may lead to a ghost peaks, baseline drift, poor reproducibility, and interfere with the analyte measurements. And also, of course, traces of organic molecule if you use to identify and quantify with accuracy what exactly you are analyzing. 60 milliliter of various water qualities were pre-concentrated on C18 column before running an acetonitrile gradients in order to see the effect of water impurities. From 2 microgram liter of organics to 20 microgram liters, there is a big difference when it comes to the noisy baselines and unknown peaks. Effect of ion contamination also has to be watched out on mass spectroscopy. For example, here we are using a fragment of peptide called bradykinin, and it infused it directly into mass spectrometer. When a small amount of sodium chloride was added to the molecule water, even with freshly prepared, when prepared with the peptide, we saw a lot of more peaks, like adduct peaks, making the interpretation of the spectra much more complex. And here, watching out for the ionic contamination whenever you are doing LCMS, not just only watching the microorganisms. So, as a summary, selecting the optimal solution for HPLC and LCMS, always use ultra-pure water with low TOC, less than 5 part per billion, optimally less than 2 part per billion, low ionic level at 18.2 mega ohm, low particles and low bacteria. Always watch the water quality, freshly purified, prepared, and with good quality of pre-treatment of the tap water feed. Specific cartridge also containing C18 silica for ultra trace LCMS analysis are recommended and are available with Merck Millipore portfolio. And finally, it's very important that we are monitoring the consumption of the type 1 or ultra pure water inside the lab, and also differentiate type 2 and type 3 water when it comes to the application. We have a huge portfolio starting from 5 to 300 liter per day requirements of type 1. Feeding the water system is also depending on the quality. So if you have a source of pure water, it can be upgraded to ultra pure water, or we have also systems taking from tap water directly to the ultra pure water. For the labs which is requiring high consumption of grade 2 water, we can also offering a very specific large capacity systems for type 3 and type 2, which can be distributed inside the building for different labs inside. Finally, thank you very much. And please, whenever you are having questions, we will be happy to answer your questions. And thank you very much. I believe now we can take some of the Q&A questions. Stanley?
Yeah, thank you, Vivek. Vivek, thank you, Ahmed. We have some questions already in the Q&A chat. The first one is coming from Joseph Olamuju, and this question is directed to Dr. Vivek. It says, for high particulate sample, is centrifuging preferable?
So for high particulate sample, you can centrifuge your sample and collect the analyte or the top layers of solvents. What happens is if the particles are not completely settled down at the bottom, or if they haven't formed a good pellet at the bottom, when you try to take the supernatant, you might disturb those particles, and those particles can get back into the solution again. So for particulate solutions, it's preferred to use a syringe filter rather than centrifugation.
Okay, thank you very much. The next question is coming from Adewonuola Adebimpe, and this question is directed to Ahmed. You say, why is degassing necessary with ultra pure water before HPLC analysis?
Actually, dissolved gases is always creating some problems when it comes to the high pressure or high back pressure, especially if you look at the injector. Whenever you are having a small air bubble inside, it will create also a fluctuation in the pressure, which is affecting the performance of the eluent and also the sample retention time, and as well as the performance of the interpretation of the results. So it's always important to have removing the dissolved gases before using the purified water as a mobile phase.
All right, thank you very much. This question is for Dr. Vivek, coming from Olufemi Adebola. He says, sir, how can I determine the particle size of the sample before filtration in order to select appropriate filter?
So the particle size that you have will be dependent on the type of formulation. If you are working in pharmaceutical, it will depend on how fine the API you're grinding or when you're mixing with the excipients. So that means you need to have that knowledge about particle size of your analyte. Typically, a 0.4 or a 0.2 micron membrane is sufficient for HPLC or UHPLC analysis. But if somebody is working on nanoparticle formulation where the size of their particles is in nanometers, they might have to go down to a 0.1 micron membrane, which is 100 nanometers in size. And if they have even smaller size, then they might have to go down to an ultrafiltration membrane, which are generally have much lower pore size than the microfiltration or the typical syringe filters. So that's how you'll go select filters.
Okay, thank you very much, Dr. Vivek. The next question is coming from Bamidele Rashid. It says, why do we still have just a minute. Why do we still have deep water peak? Why using ultra pure water in HPLC analysis? This question is directed to Ahmed.
Sorry, Stanley, would you please repeat the question?
The question says, why do we still have deep water peak while using ultra pure water in HPLC analysis?
Deep water peak?
Probably maybe the result is not coming out.
It's depending on the type of contaminants is coming from the water he is using. As we said, we are speaking about 5 types of contaminants, and some of them can be interfering with the sample, which is also creating some unpreferred or ghost peaks, which are unexpected. So always we need to purify the water to the type 1 whenever we are working with the HPLC system, so that we can also define what type of contaminants that is creating these deep peak. It could be, as we said, sodium chloride, it could be silica, it could be kind of contamination that we need to remove before using this type of water in the HPLC.
Okay, thank you. This one is coming from Ade Wonola Adibigbe. The question is directed to Dr. Vivek. He says, do you have to filter to waste each time you use a filter if using multiple syringe filters due to the sample having high number of particles? Dr. Vivek, the question?
Yeah. So when you are filtering a sample containing high level of particulates, if you know that the membrane that you are using has higher level of analyte binding or extractables, the only way to get rid of analyte binding or extractables is by flushing the membrane or syringe filter with your sample. So unfortunately, if you are going to use multiple syringe filters, and if you know that the membrane has analyte binding, you have to flush the membrane before you can collect sample for downstream analysis. What I would recommend in that case is starting with a hydrophilic PTFE membrane, which is known to show lower level of analyte binding and extractables, so that you don't have to then waste or flush your sample every time. Or going to the multi-layer filter that I talked about in the seminar, where you can use a single syringe filter to get a larger volume of sample filtered. During method development, you should always be looking at the analyte binding and extractable characteristics of the membrane that you are choosing with the sample or the analyte that you are interested in. Because as I said, it is dependent, both the analyte binding and extractable are dependent on the type of analyte, the chemical characteristics of analyte, and the chemical characteristics of the membrane.
All right, thank you, Dr. Vivek. Ahmed, this next question is for you. He said, please, what is the difference between pure water and ultra pure water?
As we said, the quality of the type water, a type 2 water starting from 1 mega ohm up to 10 mega ohm. Ultra pure water, we can link it to the type 1 water, which is 18.2 mega ohm resistivity, and it will have a TOC level less than 5 part per billion, and bacteria should be less than 1 colony forming unit per 10 milliliter. And here we can speak about ultra pure water. Pure water, as we said, it can be type 2, and it can be even type 3 water. So defining the water quality is the main issue, is a key standard to follow the standard which we are using inside the lab. When it comes to American Society for Testing and Material like ASTM, they are speaking about 18.2 mega ohm for the highly purified water. When we are talking about ISO, they are speaking about 10 mega ohm. So we need to define the water quality we are using inside the lab. Ultra pure water, mainly relating to the type 1 water, which is 18.2 mega ohm.
Right, thank you very much. The next question is coming from Vipul Desai. Is there any experimental methodology to determine analyte binding to filter media and extractable from filter media? This question is for Dr. Vivek.
Yes, so the way you would determine analyte binding is the experiment that I showed in one of my slides on the seminar. So you have your sample, and that is filtered through the membrane, and you can collect various fractions of the filtrate that are coming out, and you can clearly see if there is analyte binding to the membrane or not. And extractables, again, you do a very similar experiment. What you do is, if you are looking at extractables from a specific solvent, you can filter that solvent through the membrane and collect various fractions coming out of the filtrate and analyze them by HPLC. And you can clearly see that the extractable levels start to go down as the volume fraction keeps going up. And similarly, you'll also see that the analyte binding goes down as the filter gets more and more saturated. So that's why typically we recommend that if there is a concern about analyte binding or extractables, a customer should discard roughly about 3 to 5 ml of sample before collecting any sample for analysis. That generally takes care of extractables and analyte binding.
Okay, another question for you, Dr. Vivek. This is coming from Declan Ngoji. He said, what caused the downward trend in the peak during assay below 0? Also, why change in wavelength during a particular assay?
I'm not.
Question?
Can you repeat the question again? Not
what causes the downward trend in the peak during assay below 0? And also, why change in wavelength during a particular assay?
So the downward trend, I'm assuming that they are asking about the baseline in an HPLC. Probably. And so the baseline in an HPLC system can drift or change or go downward or upward depending on the solvents that you are using or the solvent combinations that you are using. If there are impurities that are present in the solvent, that can cause the baseline not to be stable. And the wavelength, I'm not very clear because typically in a HPLC analysis, either you are using a single wavelength for detecting your peaks of interest, or if you are using a scanning detector, scanning UV detector, then you're using a wide range of wavelengths going from 200 to say 400 nanometer. So I'm not very sure what they meant by changing wavelength during the analysis.
Okay. Another question for you, Dr. Vivek. We have roughly about 3 minutes to go. Please, can you kindly shed light on how to best determine a suitable solvent for an analyte of interest in a sample product?
So the appropriate solvent for analyte of interest is going to be based on its physicochemical characteristics. That is its log P or hydrophobicity, its log S or solubility, its pKa, which means its acidity or basicity. And many of these parameters are available in literature, or there are chemical softwares that can provide you a guidance. But then the actual solubility is determined by experimental methods. You have to try out various solvents and solvent combinations, as well as mixtures of salts or buffers, and you would determine the solubility of a compound.
Okay, one more question for you, Dr. Vivek. This is coming from Olufemi Adegbola. He says, sir, how can I determine the particle size of the sample before filtration in order to select appropriate filter?
So again, this particle size determination, it depends on the processing of the API or the drugs that you are doing in order to get to the final particle size, and then the particle size itself can be measured by microscopy, or there are laser particle measurement systems that are available, which will give you a rough idea as to what is the particle size of the particles of your analyte to choose the right membrane size.
Thank you very much, Dr. Vivek. Ahmed, there's a question for you from Ugochukwu Kezie, and it goes thus: are there specifications as regards the type of bacteria permissible in purified water?
Actually, there is no main bacteria nominated in the standards, but all types of bacteria are not allowed to be inside the water while we are using HPLC, because bacteria is also producing some products like the enzymes, proteins, and even nucleases, which is also not preferred to be used during the HPLC analysis. So we are having this 0.2 micron filter and also the UV at 254 nanometer in order to remove and kill bacteria during the purification stages of the water. So there is no chance to have any type of bacteria when we are talking about type 1 for HPLC.
Okay, last question, and this also is going to you, Ahmed, coming from Wasai of Faduba. It says, analysis of chemical reference standards using distilled water 2 microsiemens has given me a good chromatogram with a single peak, void of impurity peaks. Is the ultra pure water still useful? And how can we obtain the ultra pure water?
Actually, when we are talking about distilled water, we are talking about very old technique of water purifications. And actually, the quality of the distilled water doesn't have a real number because it has a range between 0.4 up to 2 microsiemens. These distilled water are also under the risk of recontaminations due to the many contaminants like the atmospheric contaminations and also the absorbance of the CO2, which may also change the pH as the distilled water. So I'm not recommending at all to use distilled water in the HPLC because of the risk of the recontaminations. When we are talking about the ultra pure water, also we need to be speaking about better results when we are looking at the HPLC chromatogram, and also we can grant the reproducibility of the readings of the results using a consistent water quality of 18.2 megaohm against the distilled water, which is not having a consistent water quality. It has a range of different number of qualities. So she will obtain much better results, consistent, when she's going to use ultra pure water of 18.2 megaohm.
Thank you very much, Dr. Vivek and Ahmed, for your answers. Unfortunately, we are pressed for time. We still have a lot of questions in the chat box. We are actually going to reach out to you with answers to all those questions through the email addresses you've provided. So at this time, I will hand over to Dr. Olubukola.
Thank you, Mr. Stanley. Thank you.
Thank you, Doc.
I'm sure it's been a very interesting time. Yes. And this brings out that we all have so many things to take home. For me, I know that sample filtration for HPLC analysis is very important as it reduces variability and improves consistent and reliable results. Water is also a key reagent in any laboratory and definitely needs serious attention, particularly in instrumental techniques like we've had this afternoon, particularly in HPLC, to be able to obtain very accurate results. And I know that now practically we have learned, and the choices are what we now have to make from filters and the quality of water that we use. Thank you very much, Dr. Vivek. Thank you, Mr. Werida. Thank you, Mr. Stanley, all from Merck Group. We're very grateful. And I also appreciate the Bloom Public Health team. I know that you're out there. Thank you. And to all the participants, thank you for your attention and allowing us into your space. It's been a wonderful time. Thank you very much. And to let you know that another webinar comes up in another 2 weeks, it's going to be very, very exciting time again. So please stay tuned and watch out. Thank you.
Thank you very much. Thank you.
Thank you.
Have a good day, everyone. Bye-bye.
Thank
Bye.
you.